any small amount of acetylcholine kills the ripple. Any small amount of norepinephrine kills the ripple. It's because all these neuromodulators work on potassium currents and so on. They prevent synchronization. They prevent working togetherness or working together of the whole point of these subcortical neuromodulators that they try to make independent computations as much as possible. So if you look at a isolated hippocampus, in my previous slide, we transplanted embryonic hippocampus to the adult hippocampus in either isolation or into the hippocampus itself. And we showed that the hippocampus in isolation does nothing else, just sharp wave, nothing, sharp wave, nothing, sharp wave.
It's like a default.
It's a default. And that's exactly what happens in the neocortex. The default is up and down state, up and down state, up and down state. And neurons burst, pause, burst, pause.
Okay.
They finally get.
So this is why non-REM sleep looks the way it does on an EEG. In the absence of all those neuromodulators, you get this slow oscillation of up and down states.
Exactly. So in order to take out the neurons from that enslavement, then you need subcortical neuromodulators. So when the animal is stopping, because the animal's goal is to reach something. It's always the brain generates actions towards a goal. Once the goal is reached, the animal stops, eats, drinks. It's called consumatory states. At that time, subcortical neuromodulators drop and allow the emergence of sharp wave ripples.
So sharp wave ripples are not only happening during certain phases of sleep. They can happen during waking when the animal is in a consumatory state.
Exactly. Exactly.
So when you've reached a goal, you've gotten the prize, the food, the water, the comfortable bed, that is the context in which the sharp wave ripples normally come about.
Yes. And these waking sharp waves, we just had a paper about this recently, these waking sharp waves mark or tag the important aspect, what led to this result or led to the success for future consolidation that comes a little bit later during non-REM sleep.
Yeah.
So every single night, humans have about 2,000 to 4,000 ripples. So you have 2,000 to 4,000 chances of consolidating snippets of the waking information.
Is that mostly during non-REM sleep or a certain part of sleep?
Exclusively during non-REM sleep, compressed mostly during deep stage 4 sleep. The
deepest phases. So people who have a disrupted sleep pattern or circadian biology are going to not be going to that deep phase and therefore not getting the sharp wave ripples. And that there's a clear line you can draw from that to the glucose result and how it might connect to metabolic dysfunction.
Not only them, including me, all the persons, who don't have stage 4 sleep with the old terminology. You don't have slow wave sleep, and the number of ripples are fewer. The growth hormones are not released properly, and so on. So we are much more prone to metabolic challenges.
And there's no good way to reinvigorate that sharp wave ripple tendency?
Yeah, of course, there is a way, and we have had a paper about that. We can prolong sharp wave ripples, and we showed that the cognitive ability of the animal increased.
How do you do that, though?
So what you can do is optogenetically, you can activate neurons